Our next speaker is Amos Gutnick, and the title is Implementation of Upstream Mitigation Strategies to Minimize Residual Plasmid Backbone and Host Cell DNA Encapsulation in Recombinant AAV Vectors.
Thanks a lot. Thanks everybody for showing up for this morning session. I think we're just past the halfway point in this great meeting, so I want to thank the organizers for a terrific conference and for this opportunity to share with you guys some data that we hope will be helpful. My disclaimer is I work for PackGene Biotech. If you're unfamiliar with PackGene, we are a global CRO and CDMO in the AAV space. We serve customers from small academic labs to giant multinational pharmaceutical companies. Since 2014, we've developed dozens of technological innovations, and there's always a laser focus on lowering the cost of manufacturing through increased efficiency, increased purity, increased yields, in order to make gene therapy affordable. Our CDMO capacity is fairly significant, as you can see here, and is located in two twin facilities that we have in Houston, Texas, and in Guangzhou in China. And I say twin because they share SOPs, instrument analytics, they share expertise, and so that seamless tech transfer has been very valuable to a lot of our customers, especially lately with a lot of companies trying to navigate regulatory landscapes on both sides of the Pacific. So I mentioned that just because there has been a lot of talk in this meeting this year about the IIT pathway for de-risking path to the clinic, and that's been something that we've really focused on with our unique global footprint. So today I want to talk to you about ways in which we've found that small, simple redesigns of our plasmids can have great benefit for cleaning up mispackaged non-vector DNA. So invariably, AAV preps carry non-vector DNA from the production plasmids, and this is problematic. Of course, as low as we can keep it, there's still always a risk of immunogenicity being higher in the prep, off-target expressions, even insertional integration into patient DNA, and of course, reduced potency at any given titer. So obviously, the regulators want to keep this as low as possible, but is it low enough? That's not clear. Just to hammer the point home, if you're sitting here, you've probably seen this figure from earlier this year from the Brewer lab. This is a staining for bacterial origin of replication sequence from a plasmid backbone, and it's in the liver biopsy of a patient nearly 7 weeks after she was treated with Zolgensma. So this is a pretty stark reminder that we still have a ways to go to try to lower this non-viral packaged DNA, and certainly I think the regulators think so as well. So how does an origin of replication sequence end up encapsulated? Back to the basics, which I'm sure everyone here knows, just the 3 plasmid system. So the rep complex recognizes the ITRs and therefore encapsulates the transgene. However, we know that the rep complex is not infallible. It can read through the transgene and then package some of the backbone DNA. It can even package only the backbone DNA through what we call like reverse packaging, and it makes those mistakes regularly. There are also the rep binding elements on the P5 promoter, which can mimic the ITRs, and that can cause packaging of the specific rep cap sequences or some more backbone from that plasmid. And we don't have any suspected motifs on the helper plasmid. So we ran an NGS on a standard AAV GFP prep. The vast majority of the prep is exactly what we would expect, the correct GFP transgene, but we have a non-0% of mispackaged. We have about 0.5% of the host cell DNA, which I'll just touch upon later in this talk. And we have almost 0.7% of backbone DNA. Now, we think that most of this backbone DNA is coming from the gene of interest plasmid. The reason we think that is you see there's twice as much of that as specific rep capsid sequence in this prep. However, if we run a control experiment where we're just making empty capsid, so no GOI plasmid, we see that the majority of the encapsulation is from the specific rep cap, not really from backbone. So that's why we think most of it is coming from the GOI plasmid. So how do we prevent backbone from the GOI plasmid from ending up inside the capsid? All right, so it's been known in the field for a decade now that if we, before our transfection, excise out the backbone doesn't end up in the capsid. That makes a lot of sense. Whether we do that in the bacterial preps beforehand or in vitro, if we put recognition sites flanking the ITRs and we recombine out or excise out the backbone, we have a much, much lower chance of having that backbone being mispackaged. So what we went through is we tried to go through several methods in which we could have that excision happen inside the production cells without having to add extra steps, extra manufacturing costs, trying to keep it simple. So I'll just briefly go over the first three methods that we tested. They're the protelomerase that creates these doggy bone DNAs, ISD1 meganuclease restriction. We tested CRISPR, adding CRISPR sites, and in all these cases, we ran qPCR for the origin of replication sequence. That's our proxy for the backbone, and the Whipper sequence is the proxy for our transgene to see how those ratios change. And in all of these cases, we actually get very significant reduction in backbone, which is nice. But in a lot of them, that comes with a price in titer. We get less yield. And again, then we're not really keeping manufacturing costs down, and that is very important. The fourth method really surprised us, and this is just straightforward Cre-LoxP recombinase, and we got significant reduction in backbone without any reduction in our yield. Pretty straightforward. We put LoxP sites flanking the ITRs, and we add in a fourth plasmid expressing Cre under a strong CBH promoter. The Cre recombines the gene of interest plasmid into two mini circles that are pretty stable. And we see here that we don't have a negative impact on our titer, but a huge reduction in backbone DNA and backbone carryover. Interestingly, we saw that when we put the more of the Cre plasmid we put in, we actually do get a reduction in titer. So we wanted to ask two questions. If we express less of the Cre, maybe by a weaker promoter, would that be even better? And second of all, keep manufacturing costs down, to keep the operation simple, can we take the Cre gene and put it into our helper plasmid in order to bring it back to a 3 plasmid system? So we did just that, and we were very surprised to see that on a weak promoter, we actually got 3 times the yield, which was excellent, and all of that with a very significant reduction in backbone carryover. We went back to do an NGS experiment. This is a different prep for a different transgene. And in this case, we have the LoxP sites in there, but without the Cre and the helper. We have those two species of contaminants. But if we swap out that helper for one that has a Cre on it, we've almost completely resolved, I would say, the presence of backbone DNA. And this is a very simple redesign of the plasmid. So what about this host cell DNA? I think the evidence is mounted that what is happening there is that the stress of the transfection is causing apoptosis in the production cells. And so it stands to reason that if we can somehow inhibit the caspases, that we would be able to prevent the apoptosis. So yeah, we went ahead and just screened through a lot of available capsase inhibitors, and we were happy to see that two candidates from a similar class relatively low concentrations in the media. They don't hinder our yield, but they almost completely get rid of host cell DNA. So in general, I've showed you how we have systematically gone through to try to find viable, economically sensible solutions to increase our purity of our product without increasing manufacturing costs, without adding more logistical complexities. And now we're working obviously on scaling this up to see, how we can do this to benefit material that goes into the clinic, into clinical trials for the benefit of our partners and for the whole field. All of this data, we posted it up, so you can feel free to look it up online. I will just say that if you have any further questions or you want to discuss, you can ask now if there's time, but you can visit us at our booth, and we're happy to discuss this and some of our other technologies and our capabilities. There's also some cool swag. There might be some more AAV core Lego sets there if you hurry. And yeah, that's what I have to present today. Yeah, any questions?
Thank you very much. Do we have any questions? Yes, I see the first one.
Hey, great talk. When you were assessing yield impacts of changing the plasmids, did you do anything with the transfection optimization of the parameters, like the ratios, or change this at all?
So when we moved from a 3 plasmid system to a 4 plasmid system, then obviously that changes the ratios. But adding into the helper, we didn't see any significant need for changing the ratios between the plasmids, and that didn't affect the yield at all.
Got it. But when you removed the backbone, did you have to do any optimization?
Oh, we didn't have to do any optimization. I mean, so the first 3 methods that I showed, those end up with a linear plasmid for just the ITRs and transgene. But when you do the mini circle, it's very stable. And so, yeah, like as you saw, we didn't have to re-optimize it, and we actually got a 3-fold increase in yield, probably just because the increase in stability and maybe accessibility to the rep complex.
Great, thank you.
Sure.
Next question, please.
Hi, just a quick question about the sequencing. Was that short read or long read sequencing to detect the contaminants? And the reason I ask is because in my experience, like short read is definitely more sensitive for picking up what contaminants are in your prep than the long read.
So that's a great question. I think that it was short read, but I do want to caveat that I might be wrong. So, so if that is the case, I would say definitely look at the source that I gave you, or come by and I will definitely look that up. I think that's an important point. I just don't want to commit to that. I think it was short read though.
Yes, please.
Great talk. One question on the caspase inhibitors. So do you check for the caspase inhibitors in a final drug product, any residuals or any other untoward effects of these inhibitors on the vector purity?
Yeah, that's an important point. That's part of our current scale-up efforts, I would say. So I don't have a definitive answer yet. That's certainly something that we're interested in resolving to make sure that this could scale up into the clinic. But right. I think it's an important point.
Another follow-up question on that, right? Do you see, I believe that you have done some basic small-scale downstream purifications on these. Do you see any effect on the empty-full ratios or anything due to these inhibitors that you add?
Not that I'm aware of at this point. Again, this is ongoing research. So at this point, I'm not aware of any significant reduction in empty-full. But I think that as we try to scale this up, that might change. So that's certainly something that we're looking at. Hopefully we'll have some data next year.
All right. Thank you.
Thanks.
Next question.
Hello, thank you for a highly informative talk. I had a question about one of your earlier slides. You showed, I want to say, roughly 75% of mispackaged DNA was coming from the RepCap plasmid.
Right.
And you said then you actually took the empty band and analyzed that. And that was, I guess my question is, why is
Try to
it
go
the
back.
empty band? Was it maybe convoluted with the intermediate band? Just trying to gain clarity on that.
Yeah, I think that's a great question. I'm trying to cycle all the way back here.
Yep.
So
Yep.
yeah, so this was when we were running, we were making empty packaging. So this prep is 2 plasmids, no GOI. So that's why in this case, there's obviously a lot less DNA being packaged at all. This is mostly empty. But from the DNA that has been packaged, 75% of it turned out to be from the RepCap sequences. So I think that this isn't showing how much DNA is being packaged, just the ratios. And really, this was a way for us, a sanity check, to see how much of the backbone we think is coming from that plasmid compared to the GOI plasmid. And I think it wasn't a lot. We've already done a lot of modifications on our RepCap design in order to minimize it. And so I think that that was really the point of for us of running this empty capsid iteration.
Okay, thanks for clarification.
Yeah.
The last question, really quick.
Yes,
Quick answer, please.
is following that the whole cell DNA that you have with the CRE system is higher than when you don't use it. Do you see some toxicity when you express CRE?
Well, do we see toxicity?
Yes.
We do not see toxicity. It's possible that, we did see that when the more CRE we express, we get a lower yield. It's possible that that is a result of toxicity. I don't know if we
Because it's a nuclease, it's common.
Exactly. So I think that might be the reason why the ability to modulate the expression by using a much lower promoter has been so useful to us. We've also separately tried to incorporate that into the CRE into the cell line, and we've got similar results as long as we're using a very weak promoter. So I think that that's key to preventing that toxicity.
We have to move on.
We'll talk later.
Coffee break.
Okay. Coffee break, everybody.
So thanks again.
Thank you.
We have some spaces here. So for those who are standing in the back, we have still some seats here. And then we move forward.